Dr. Li Wong is with the SUNY Upstate Medical University in Syracuse, New York, and we're very excited about her project that's looking at the impact of the exercise hormone irisin on tauopathy. She will be awarded a $100,000 CART grant. Dr. Wong.
My project is alternative splicing mechanism in Alzheimer's disease. And, this is really thankful for this grant. And I'd like to give special thanks for Dr. Gofford for his help during this application process. This is a very new project to my lab. Generally, I focus on chronic stress and comorbid conditions like major depression, anxiety, obesity. And so this is a first to my lab into two subjects into alternative splicing, which is a totally new avenue for us, and Alzheimer's disease. And I'm very excited to start or continue this line of research. And I am a professor and co-director of the Precision Medicine Laboratory in Psychiatry at Upstate Medical University. Giulio Licinio is my co-PI and co-PI, co-director and co-PI in this project. Dr. Lu is a friend, and also she has helped me generate some Alzheimer's disease data here using animal models. And Dr. Ma and Juan, they are co-investigators working in my lab for about 2-3 years and help me generate the pilot data that I'm gonna present later on. What is alternative splicing? I would say I need to go back to the basics and present this, or actually I think refresh our memories into the central dogma of biology, which is you have one gene, DNA, and that DNA generates one RNA, and then that RNA generates one protein. And, this has been going on for a long time, and of course, nothing stands still, and this has expanded. In reality, what happens is that we have one gene and multiple messenger RNAs that through a process called alternative splicing, one gene can generate several proteins because of this process here. After the sequencing of the entire human genome, it came really to a surprise to all the investigators that humans actually did not have many more genes than a fly. And but how can that be? Because we are so much more complex than flies, And how can we achieve the proper development and physiological functions if we don't have lots and lots of many genes anyways? What happens to our genes is that they encode multiple messenger RNAs, and through a process called pre-mRNA splicing. And this process occurs predominantly in the brain, but happens in all the tissues in the body. And these protein isoforms can have similar functions or different functions. And for instance, look at the gene that we have all been focusing in Alzheimer's, APP, amyloid precursor protein that generates the beta-amyloid. It actually is alternatively spliced. So, some exons are alternatively spliced from the gene, and some proteins are different. And some proteins, some APP forms are very low in the brain, other forms, they increase in the frontal cortex with Alzheimer's, and the predominant form in the brain is called APP695. So, this is, we know this, and this is going on for some time. The science in terms of alternative splicing has been advancing. But how this process is relevant to aging. Now we know that alternative splicing increases with aging. So the process of alternative splicing varies from in terms of during the lifespan. And here I have 6 examples of genes that you can see through is gene expression through lifespan. And you see that most of them increase, most of them are low here and during young adulthood, at the childhood. And then at age, when we start aging, it starts to go up in the elevation. So they are alternative spliced. And as we age, the number of alternative spliced genes in several tissues increase, including in the brain. So, and this paper actually exemplifies a lot what happens in Alzheimer's disease, how alternative splicing is very relevant to Alzheimer's disease. Because in Alzheimer's disease, there are several missplicing events in the brain. And these events are actually associated with beta-amyloid burden and neurofibrillary tangles. This is a complicated thing. I'm not gonna go over, but there's several genes here and 450 participants with really a very large study showing that, there are alternative splicing events that are going on wrong in Alzheimer's. And what aspects of alternative splicing will this project focus on? Alternative splicing is performed by a little machine in the body called the Spliceosome. This machine is very complicated, has more than 100 proteins, and has several steps and stuff. And so, it would be impossible to study all the elements of this spliceosome. So this project focus specifically on the Exon Junction Complex. And what is this complex? This little complex here, it's tags itself to the what they call precursor messenger RNA, and then to a specific region, which is gonna be spliced out of the mRNA. And then so will give you rise to splice variants. And this complex is formed by these 4 elements. And these elements, we are going to specifically focus on this molecule called CWC22. This molecule is essential for the formation of the EJC function complex, because without this molecule, this other molecule here, which is the main element of this complex that then will assemble the rest of the complex, will not be going to the spliceosome. So this is a chaperone that drives this molecule into the spliceosome. It starts this deposit of this EJC function into the spliceosome in the messenger RNA. And why are we focusing on this specific factor? This is an essential molecule for the splicing, uh, alternative splicing of genes, and it's highly expressed throughout the brain. And we found out that it is modulated by chronic stress. So, when there is chronic stress, the animals that are chronically stressed have lower expression of the CWC22. And CWC22 localizes primarily in the nuclei where the splicing, the alternative splicing process starts. And so there is also a mislocalization of this factor into the cytoplasm with chronic stress. Then we also found out that overexpressing this element decreases dendritic length and branch and synaptic strength. So it is really involved into the synapses, healthy synaptic of neurons. And when we decrease this gene, the number of dendritic spines and memory spines is increased. So too much seems to not be doing very well for the neurons and the synaptic function, and decreasing it seems to improve your memory spines and electrophysiology function. And we also found out that CWC22 expression increases in aging and Alzheimer's disease. So here you can see mice of different ages. And age 22, there is an increase of CWC22 expression. This is hippocampus. And in the 5AD model of Alzheimer's, it's also increased in the hippocampus. And this age of, when the CWC22 messenger RNA is increased is when the animals, actually the normal animals, have a decline in cognitive function in terms of two tests here that test immediately recognition memory location and novel recognition object recognition test. So my project is to understand if alternative splicing is a potential target for Alzheimer's disease treatment. And if addressing aberrant alternative splicing mechanisms may improve Alzheimer's disease treatment. So I will overexpress this gene in young mice and see if I can accelerate brain aging or Alzheimer's disease progression. As you can see from my data of expression, really impaired synaptic function of neurons. And the second aim will be understanding if hippocampal decrease of CWC22 will rescue Alzheimer's disease associated brain changes. And we use tools developed for Alzheimer's disease, like the animal models, electrophysiology, neuropathology, cognitive tests in these two experiments to understand the impact of this specifically alternative splicing component will change brain function or pathology. And so we hope that this project will generate data to help understand if this alternative splicing, aberrant alternative splicing, would improve Alzheimer's treatment. And if modifications of expression of Alzheimer-associated genes could play a role in disease prevention and therapies. In reality, there are clinical trials right now in cancer and spinal muscular atrophy. these two diseases are known to have alternative splicing aberrant or deregulated. And so there are clinical trials with new compounds that interfere with splicing, RNA splicing, and they are just underway. And I hope that, we can find some way to understand the process of alternative splicing in aging and Alzheimer's. And thank you so much for your funding.
Well, let's give you a virtual check.
Thank you.
So Dr. Wong, we're very, very happy to present you with a check for $100,000 in support of your very important research. Thank you so much for being one of our CART recipients this year.